Okay, we will now start the presentation of Desmopressin. My name is Yasmin Mataman, and I will start with the introduction of what is Desmopressin and the usage of Desmopressin. Okay, first, what is Desmopressin? Desmopressin is a synthetic hormone that is usually administered in the form of its hydrated acetate salt, which is desmopressin acetate, and is used to replace vasopressin, which is more known as ADH. Next, the usage of desmopressin. First is for diabetes insipidus, which is a condition the body produce an abnormally large amount of urine. Then it can also be used to control excessive thirst and the passage of an abnormally large amount of urine that may occur after head injury or after certain type of surgery. It can also be used to be in nocturia, which is a condition that causes you to wake up during the night to urinate. Last, it can be used to cure mild hemophilia A, which is a hereditary bleeding disorder caused by lack of blood clotting factor 8. Continuing with the previous slide, desmopressin can replace. What it actually do is it replace vasopressin, which is ADH, which is a hormone that is normally produced in the body to help balance the amount of water and salt. So the more desmopressin one consume, the less urine they will produce. Another side effect is they can increase certain clotting factor that can treat some bleeding disorder like mild hemophilia A. Moving on to the chemical structure, as you can see, we have 46 carbon, 64 hydrogen, 14 nitrogen, 12 oxygen, and 2 sulfur. The molecular weight average is 1069.22. Monoisotopic is 1068.4270.
Okay, my name is Nur Nadia Adaha binti Muhammad Yusof, and I will present how to synthesize desmopressin. Desmopressin being produced by solid phase peptide synthesis method SPPS using the cyclization of the peptide in resin. Here are the pictures that important in SPPS. First picture is FMOG or bulk is a special protecting group which will use to protect amine functional groups from reacting when we don't want them to. Second picture is solid phase. Solid phase synthesis is a powerful strategy for multi-step reactions, and solid support is a plastic polymer functionalized with a linker molecule, and these are often referred as resin. We will synthesize our molecule at the surface of a solid polymer support. This allows us to isolate the product that we want out of each step without having to go through many isolation of the product using other methods. The solid phase strategy saves a lot of time and dramatically improves to yield the product. So the third amino acid or the peptide that we want, we will create the peptide such as a long chain or a polymer of amino acid. So we will perform simple carbonyl substitution reaction during the synthesis. As you can see at the diagram, we have peptide synthesis cycle. First, we have the resin, and we undergo the deprotection process, and we activate the amino acid to go under coupling reaction. So we repeated the steps until we get the long chain, and then we cleavage and we get the wanted peptide. So first, the solid support. The solid support, as we know, is a resin. There are many options to choose for when selecting the solid support for solid phase synthesis. The example of resin is ring amide and BHA resin and others. So second is a deprotection process. So before adding glycine, the FMOG group must be removed. So how to remove the FMOG group? is FMO is stable in acidic conduction, but it is easily removed by a weak base due to the acidic hydrogen in the FMO structure. So we use piperidine as the base for the deprotection reaction. So the third one is activation of amino acid. So activating the carboxy group of a glycine residue that has its amine protected by an FMO group and its side chain protected by an acid labile group. So the carbonyl is activated using a coupling reagent by converting the anionic oxygen into an acceptable leaving group. So this is how the way of activation. So the fourth one is the coupling. So we couple the glycine residue to the D-arginine residue on the solid support and treatment for the solid support with a base to deprotect the amine group of the D-arginine. Soluble side products of the coupling reaction are rinsed away and the amino acid is left covalently attached to the resin. We need to repeat the three steps, which is deprotection, activation of amino acid, and coupling with other amino acids. So we have started the reaction with glycine and D-arginine. So then we continue with proline, cysteine, asparagine, glutamine, phenylalanine, tyrosine, and lastly mercaptopropionic acid. So here are the flow of solid phase peptide synthesis of desmopressin. As we can see, the FMO amino acids were used with the trityl and the PBF as side chain protecting groups for asparagine, glutamine, cysteine, and arginine respectively. So the couplings were done using the IC in a mixture of DMF at a ratio 1:1 in most of the cases, as the amino acids were coupled to full access. For this amino acid side chain, protective groups are used to prevent undesired reaction. In the FMO strategy, it is important that the side chain protecting groups are not sensitive to base, that these groups remain on the side chains throughout all coupling steps and do not fall off during the piperidine deprotection steps. After the peptide chain is complete, the final FMO should be removed and the N-terminus can be either left as a free amine or it can be kept with a subtle group. So we come at the final part, which is cleavage and cyclization of the peptide. So TFA is a common cleaving reagent to cleave the peptide from the resin and remove the side chain protecting groups. TFA is known as a strong acid and it will react with the linker and the side chain protecting groups, which are acid labile. The free peptide in the solution can be washed away from the resin. Next, circularizing the peptide formed by simultaneously deprotecting and oxidizing the thiol groups of the cysteine residue and the mercaptopropionic acid residue by contacting the solid support with an oxidizing agent. And then the disulfide bond will form between the cysteine residue and the mercaptopropionic acid residue to form a cyclic peptide, as we can see at the diagram. Lastly, contacting with the solid support with an acid, so the amino acid side chains are deprotected and desmopressin is released from the solid support. So the diagram at below, here are the final product of desmopressin that we want. Okay, I will pass my presentation with my other friends. Thank you.
So let's go to the third part, which is also the last part of the slide, which is the complement and some of the conclusion. So we listed down a few of the facts regarding desmopressin. The first is the side effects of desmopressin on human body. So one of the major side effects that desmopressin has on the human body is called hyponatremia, which is the more you consume about the desmopressin, the higher chance you will absorb more water. So while the increase of water absorption happens, the amount of the flow of the blood gets higher, which means in the meantime, the salt contents or the sodium ion concentration in your blood of your body will decrease. The imbalance of distributing the water and the sodium ion will cause some of the serious syndrome or symptom, which we will discuss a little bit on the later slide. Those symptoms are quite difficult to deal with, and some of them are even very deadly to human body. And second is how desmopressin work as a medication. So desmopressin exerts its hemostatic effect by inducing synthesis of the 1 Willebrand factor by endothelial cell, stimulating release of the 1 Willebrand factor from its storage sites in endothelial cells and cleaving the large 1 Willebrand factors multimers circulating in plasma into smaller multimers. So next we have why desmopressin can replace vasopressin in treatment of nocturia. So basically, desmopressin has around 10 times the antidiuretic action of vasopressin, but surprisingly, 1,500 times less vasoconstrictor action. It shows selectivity for antidiuretic over vasopressin effects, and it will lead to a more powerful and longer-lasting antidiuretic action than vasopressin. That is the reason why it can be perfectly replace vasopressin in some of the treatment. Finally, we have the safety issue. So one of the most important things is still the level of the sodium ion in human body. There is very little chance that it will cause the lower level of the sodium in human body, but it's very serious because it will lead to a lot of syndrome, such as headaches, confusion, muscle cramps, some of the severe weakness, hallucination, loss of coordination, and vomiting. So it's better to consult with your doctor to get some of the advice or instruction before you consuming this kind of medicine, or better run some of the lab tests, including urine test or sodium blood level test. These are best recommended. We have some of the pictures showing the different forms of desmopressin. It's usually combined with acetate to be in the medication. So the most common one is the oral tablet that can be directly consuming from the mouth. Or we also have the injection form, or some even less common to see but also existing, which is the spray. These kind of products all have very good effect on treating some of the disease we just mentioned before. So finally, it comes to the conclusion of the project. To give a definition of desmopressin, it is a synthetic hormone, and it is usually used to treat some of the disease such as diabetes, bedwetting, and nocturia. And it is usually found in the form of the desmopressin acetate, but it also has various shapes like tablet, spray, or even injection. And secondly, there's one method called solid phase peptide synthesis method that is normally used to synthesize desmopressin. And desmopressin has a very major side effect, which is it may lower the concentration of the sodium ion in the blood, and this is usually very lethal to the human. So better to consult your doctor for more information on how much you should take or when you should take desmopressin for treatment. With this, we conclude our slide on desmopressin. Thank you for watching and listening.